Hello children. So let us further this chapter. So far in the plant tissue we have learnt about meristematic tissue. Meristematic tissue that can divide, and those dividing cells work as the reservoir of cells so that wherever in the plant body cells are required, meristematic tissue cells can divide and provide those cells. Today we are going to talk about simple permanent tissue. So right now we are in permanent tissue, and in this lecture particularly I will be talking about simple permanent tissue. So what is a permanent tissue? Let us understand this. Tissue is a group of cells which are functionally specialized for a particular function, and they are structurally in most cases alike. And permanent means that these cells will no longer divide. If they no longer divide, then what is their purpose going to be? Well, they are now destined and dedicated for a particular purpose, and for that they are structurally modified also. Now this process of getting structurally and functionally modified and dedicated for a particular function is known as differentiation. So meristematic cells only keep on dividing. They are not differentiated. They do not have any particular kind of cellular thickening or specialized structures. But once the cell has been destined for a particular function, it has to change itself according to that. So if it is going to be a strength provider, it will change its structure accordingly, and so on. So permanent means that the cells are differentiated and they have certain structural specialities. So if all the constituent cells of permanent tissue are of similar type, then we call it simple permanent tissue. So here simple means that all the cells of this kind of permanent tissue will be similar in their structure and of course their function, because they are part of a tissue. So simple permanent tissue, children, is of three main types in plants: parenchyma, collenchyma and sclerenchyma. So parenchyma cells are simplest in structure. How? They have thin cell walls, no spatial thickening or deposition on their cell walls. They have little intercellular spaces, and they have various functions in the plant body that we are going to see in a moment. First, understand that this is the most abundant and simple type of simple permanent tissue. Its cells are not highly specialized. Now, if parenchyma cells have chloroplast, then it is a subtype of parenchyma which is known as chlorenchyma. If parenchyma cells are absent from in between, leaving air spaces behind, then this subtype of parenchyma will be known as aerenchyma. But the common simple features remain the same, that thin cell wall, little intercellular spaces and no specialized depositions. The second simple permanent tissue, collenchyma, and the third simple permanent tissue is clarenchyma. And since they are permanent tissue, none of them have the division capability. So let us learn about them in some detail. So this question often comes, children: define or explain what do you understand by a simple permanent tissue. So tissue is a group of cells having similar function. Permanent means they can no longer divide, and simple means all cells are structurally similar. So what can we conclude from all of this? Well, we can conclude that simple permanent tissue is made up of similar type of cells which are differentiated for a particular function. This differentiation part indicates that they are not going to divide anymore because they have taken a permanent function and structure. So, types of simple permanent tissue are parenchyma, collenchyma and sclerenchyma. So, parenchyma. What do we need to know about this? This is the most common simple permanent tissue and most abundant also, and it consists of relatively unspecialized cells, as I have said before. These are living cells and they are loosely arranged. They have large intercellular spaces between the cell that gives further softness to parenchyma tissue. Now, where do we observe this kind of tissue generally? It is mostly found as ground or packing tissue, and it generally stores food also. So children, in plant body where non specialized tissue are required, like we neither need epidermal tissue nor do we need vascular tissue, some gap filling or packing tissue is required, then that is going to be your parenchyma. Sometimes when parenchyma contains chlorophyll, then it becomes chlorenchyma. For example, this kind of chlorenchyma tissue is found in leaves and all the green parts of the plant. And in aquatic plants, children, where we need buoyancy, so we need some air to be trapped in the plant tissue. And to do that, some cells from in between the parenchyma tissue get dissolved, and that space which is left behind is filled with air. That air provides a lot of buoyancy, and so aquatic plants are able to float. And this kind of tissue is known as aerenchyma tissue. So let us see how a simple parenchyma tissue looks like. So here, whatever we have learned so far is visible. Thin cellulosic cell walls, intercellular spaces, so that the cells are loosely arranged, and no specialized thickening or modified structures. Suppose this cell from here had been dissolved, then here would be an air space, and that air space will have lots of air, then it will become aerenchyma. Suppose here some chloroplasts were present in each cell, that would become chlorenchyma. But chlorenchyma or aerenchyma both are subtypes of parenchyma only. And when these cells store various food items like starch, oil, protein, then it is the food storing function of parenchyma. So in nutshell, you can say that parenchyma is simplest, most abundant plant tissue which functions as ground tissue or packing tissue. It stores food, and it is also involved in photosynthesis and in providing buoyancy to the aquatic plants. Moving on to the next type of tissue, collenchyma. So these are very specialized tissue because its cells are elongated, a little elongated, and irregularly thickened. This expression can be mentioned as non-uniform thickening, non-uniform deposition, irregular thickening, or irregular deposition on the cell wall. Now what deposition are we talking about? It is the deposition of cellulose and pectin. So that cellulose and pectin is not going to be deposited all over the cell wall, but only at the corners. So that is non-uniform deposition. Now why do we need this kind of tissue? First, understand that there is very little intercellular space and thickening is only present at the corners. So what happens, children? It provides a lot of flexibility, and flexibility allows bending of plant parts. And we have observed this bending a lot because there are tendrils which are helping the plant in finding a support and climbing through it, and there are petioles of the leaf which bend easily without breaking. And whenever the wind is blowing, children, you have observed that plants are swaying along with the wind. They don't break because they have collenchyma tissue. So the main function of collenchyma will be to provide flexible strength to the plant. So it will be present in all those plant parts which are highly flexible and which do not break easily. They just bend. So it provides mechanical support. Now how does it look like? So it looks like this. You can see this green line is your cell wall, but you can see the deposition is only happening at the corners. This deposition is of cellulose and pectin. So do not forget what kind of material is depositing in which tissue. And you can see that this uneven thickening makes this tissue flexible. Now next type of tissue is the major strength provider tissue, is sclerenchyma. Sclerenchyma tissue is of dead cells. Now parenchyma and collenchyma, remember, they are made up of living cells, but sclerenchyma tissue, when it is mature, it has dead cells. Initially the cells are alive, otherwise its cells will not have any thickening on its cell wall, but slowly due to over thickening the cells become dead. Now what thickening are we talking about and how does it look like? So Sclerenchyma tissue cells are long and narrow, and the walls are highly and uniformly thickened, and the tissue is having deposition of cell wall is having the deposition of lignin. It is not pectin, it is lignin. Now these cell walls become so thick that no internal space is left behind, and therefore eventually the cell dies. Now, why do we need so much lignin to be deposited? Because sclerenchyma makes the plant hard and stiff. Lignin is a very decay resistant and hard substance. So when it is deposited, even when the cell dies, that framework of lignin cell wall remains back, and it makes that part of the plant very hard and stiff to provide strength. And why not, children? We need sclerenchyma of tissue to hold the posture of the plant vertically erect. It is all the time anti-gravity, it is all the time bearing high winds, rains and everything. So the plant needs to have some kind of strength giving tissue, and this strength comes from stiffness of sclerenchyma fibers. Now where else do we see them? So sclerenchyma will be present in hard parts of the plant like seed coats, like husk of the coconut, and along the vascular bundle also. So once I will be teaching about complex permanent tissue, I will tell you that vascular bundle are like pipes which run throughout the length of the plant body. Now that is having a very important function, and we cannot afford to collapse that pipe. So something needs to run along with those vascular bundles so that it is continuously providing strength, and that is your sclerenchyma. So in the vascular bundle, along with the xylem and phloem tissue, you will also find sclerenchyma kind of tissue to provide strength. Now, where else do we find sclerenchyma? So here we have learned that they are long and narrow, but there is another variant of sclerenchyma which is short and almost circular, but the thickening part is similar. So those smaller circular sclerenchyma are known as sclerids, and sclerids are present in the flesh of various fruits like pears and guava. Do you remember, children, whenever we are eating pear or guava fruit, we feel some grittiness between our teeth? That grittiness is because of the sclerids which are found in the flesh of these fruits. So these three, parenchyma, collenchyma and sclerenchyma, are simple permanent tissue, so that the cells are similar in structure, but they have different functions because of their different depositions. Parenchyma, very simple, no further deposition, only cellulose cell wall. Here pectin and cellulose are deposited, but only at the corners, and here lignin is deposited throughout the cell wall and highly deposited, so that eventually the cell also becomes dead. So these two are living, sclerenchyma cells are dead. This is one sclerenchyma fiber, and the sclerid is a small and circular structure having the similar level of thickening. And now with this we have learned about simple permanent tissue. Here I am going to show you some actual slides that when we cut the living plant tissue and stain them with dyes and watch under the microscope, how do they look like. So this is the structure of parenchyma. You can see there are intercellular spaces, considerable spaces, and the cell wall looks normally thickened, not much. And here, if parenchyma subtype, if it develops chloroplast in its cells, then it becomes green and performs photosynthesis. So this is your chlorenchyma. Then here is your aerenchyma. This must be the section of some aquatic plant, stem or root, and you can see so many intercellular spaces because many cells from in between have dissolved, leaving these air spaces to trap air. And here is your collenchyma. So pay attention, children. The cell wall looks thickened, but at the corners you can see the thickness is more. This is your collenchyma tissue here. Collenchyma is generally present just below the epidermis, known as hypodermis. So in the hypodermal region you can see that the corners are having more of deposition. That is very much characteristic of collenchyma. And lastly is sclerenchyma. So you see this this chemical dye which has given a stain to the cell wall is showing that at every place the cell wall is pretty thick. So sclerenchyma has high deposition of lignin, while collenchyma has pectin deposition. So these are some real views of how these plant tissue look like when we prepare slides of them and watch them under the microscope. Now let us talk about epidermal tissue. What is epidermal tissue? Well, epidermal tissue is always involved in giving covering and protection to the body of the organism, whether it is body of the plant or animals. In our body also, skin layer is our dermal tissue which covers our body, and something that covers us is always going to be protective. So in plants, the outermost surface is epidermis, and epidermis has various functions, various vital functions. So we must understand what is its structure. So epidermis is made up of a single layer of flattened cells. So these are epidermal cells. The characteristic will be flattened cells, not very thick, because epidermal layer is not only the covering layer, it also has to allow gaseous exchange. So its cells should not be very thick. They would start acting as a barrier. Now these cells are thick, but since they are protective, there should not be any intercellular spaces. So cells of epidermis are closely packed. There are not any intercellular spaces between them. Now, one more thing needs to be ensured. If the cells are so thin and flattened, then a lot of water might evaporate from them. And if the water from these cells are gone, then cells from the from below will also start losing water, and so on. So plant cannot afford this. Plants are always standing in the outside environment in the sun, and they cannot start to lose so much water. So what happens, children, in the aerial parts of the plant, by which I mean stem and leaf, not the root part. In the aerial part of the plant, the epidermis develops a thick waxy layer. Do you see that the line here is darker? This line is actually showing, children, deposition of a certain substance on the cells of epidermis. So this layer, you must have seen that leaves and young stem are very shiny in their appearance. Why? Because epidermal cells have this deposition of a waxy substance. What is this waxy substance known as? Well, that substance is known as cutin, and this entire layer, which is made up of several waxes including cutin and some complex waxes, this layer which is covering the epidermis, this is known as cuticle layer. Cuticle layer. Now remember, epidermis will be present in both roots, stem and leaves, all the parts of the plant, but cuticle will be present only in the aerial parts of the plant, which have higher rates of water loss through evaporation. Roots do not have to face that because they are underground. So root epidermal cells will be just like that. They will not have cuticle. Cuticle is only found in stem or leaf epidermis. One thing. Now another interesting structure, and before I tell you more about this interesting structure, let me tell you why it exists at all. If there is a waxy layer on the epidermis of stem and leaf, then waxy layer will not allow passage of water or even gases. So how are we going to do that? Because for plant survival, gaseous exchange and transpiration is essential. So we cannot leave all the epidermal cells exposed also, but once cuticle is deposited, then how are we going to let the gases in or out? To take care of that, there are specialized gates or openings provided in the epidermis, and these openings, children, are known as stomata. These are like guarded gates So the guard knows when to open the gate, for whom to open the gate, and otherwise keep the gate closed. So let me show you the structure of stomata in some detail, and we will see that how beautifully the nature has come up with these gates. If I show you the detailed structure of each of, so this is the stomatal view. If you take out the epidermal peel from this view and watch it under the microscope, but if you cut the section of a leaf from this plane, then it will appear like this. So in this lecture we have learnt about simple permanent tissue, including epidermal tissue and what it contains. So once more I will revise and emphasize on the point, children, that epidermis is a layer of flattened cells with no intercellular gaps. The cells are very closely packed because they have following functions in the plant body. They are the covering layer, they are the protective layer, and they have to ensure that no foreign particle like dust, viruses, bacteria enter the plant body. But at the same time, they have to allow for the exchange of gases. Now, in the aerial parts of the plant, the epidermis is covered with cuticle layer, and it also contains stomata. So remember, children, in the epidermis of roots, cuticle and stomata both will be absent, but in the epidermis of stem and leaf, cuticle and stomata will be found. One more important point here, this deposition of cuticle will be it will be much more thickened in case of desert plants, because desert plants have water scarcity. They have to conserve whatever water they have. So in desert plants the cuticle is very much thickened, and these stomata are actually present at a sunken location also. Both these modifications, higher deposition of cuticle and sunken location of stomata, reduces the rate of transpiration so that the plant can conserve its water. So this is all for today. Please revise all this. In the next lecture I will be discussing about complex permanent tissue. Thank you.